This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome. Today, we explore a revolution in biology that is overturning 50 years of scientific dogma. For decades, the neutral theory suggested that most protein evolution is merely random noise. But new research is shattering that silence. We'll dive into groundbreaking studies revealing that beneficial mutations are far more common than we ever imagined. From the gambler's ruin to the hidden power of silent mutations, join us as we uncover why the story of our DNA is not one of random drift, but a high-stakes drama of constant, active adaptation. Let's begin. Chapter 1: The 1% Revolution and the Death of Neutrality. For more than half a century, the study of molecular evolution has been anchored by a single, powerful idea. This concept, known as the neutral theory of molecular evolution, suggested that when we look at the vast landscape of our DNA and the proteins it encodes, most of what we see is the result of random chance rather than the guiding hand of natural selection. It proposed a world where the majority of genetic changes that stick around over millions of years are effectively invisible to the forces of survival. They were thought to be neither helpful nor harmful, drifting through populations like leaves on a river, eventually becoming fixed in a species simply because of the mathematical laws of probability in finite populations. This perspective was revolutionary when it first emerged in the late 1960s because it challenged the traditional Darwinian view that every tiny detail of an organism must be an adaptation honed by selection. For decades, it served as the null hypothesis for nearly every genetic study. If a scientist wanted to claim that a specific gene evolved for a purpose, they first had to prove that its pattern of change could not be explained by this background noise of neutral drift. However, a wave of recent research is beginning to pull at the threads of this foundational theory, revealing that the raw material for adaptation, the beneficial mutation, is far more abundant than we ever dared to imagine. To understand why this discovery is so disruptive, we have to look at the logic that built the neutral theory in the first place. In the mid-20th century, as scientists began to sequence the first proteins, they were struck by an incredible amount of variation. When they compared basic proteins like hemoglobin across different species, they found a steady, almost rhythmic accumulation of changes over time. This led to the concept of the molecular clock, the idea that mutations accumulate at a constant rate. But this observation created a massive problem for the biologists of the day. If every one of those thousands of changes was a beneficial adaptation, the cost of that selection would be astronomical. This historical anchor is often referred to as Haldane's dilemma. In the 1950s, the polymath biologist J.B.S. Haldane estimated that it takes about 300 generations for a single beneficial mutation to reach fixation in a mammalian lineage. In the language of population genetics, this is the selection load or replacement cost. The argument was that if a population had to weed out every individual that did not have a new, slightly better mutation, the species would likely go extinct from the sheer reproductive burden of the process. There simply was not enough room in the budget of life for that much improvement. If every molecular change we observed was an adaptation, the species would have to produce an impossible number of offspring just to maintain its numbers while the inferior versions were being killed off by selection. The solution to this dilemma was Moto Kimura's neutral theory. By proposing that the vast majority of these molecular changes were neutral, scientists could explain the rapid and constant rate of evolution without requiring a high cost of selection. In this framework, beneficial mutations were seen as the rarest of events, precious, infrequent gold nuggets buried in a mountain of neutral or harmful lead. It was assumed that for every million mutations, perhaps only a tiny, negligible fraction provided any real advantage. This assumption became the bedrock of modern genomics. It allowed us to use genetic differences to estimate when species diverged and to identify junk DNA that appeared to serve no functional purpose. As the theory matured, it was refined by Tomoko Ota, who introduced the nearly neutral theory. Ota provided a crucial bridge between the rigid neutralism of Kimura and the complexities of real biology. She suggested that the boundary between a neutral mutation and a selected one is blurry and depends heavily on the size of the population. In a small population, a slightly beneficial or slightly harmful mutation might behave as if it were neutral, because the noise of random drift is louder than the signal of selection. This middle step in evolutionary history acknowledged that the world was not just black and white, but it still maintained the core idea that truly beneficial, selection-driven changes were the exception rather than the rule. But the arrival of high-throughput technology and new experimental techniques has allowed researchers to finally put that assumption to a direct test, and the results are upending the status quo. The primary tool in this new investigation is a technique called deep mutational scanning. Instead of waiting for nature to take its course over thousands of years, researchers can now take a single gene and systematically create every possible single-letter change within its code. A landmark 2025 study by Zhanzi Zhang and his team at the University of Michigan has shattered the long-held belief in the rarity of beneficial mutations. The team conducted a massive analysis of 12,267 amino acid altering mutations across 24 different genes in both prokaryotic and eukaryotic organisms, such as yeast and the bacteria E. coli. By creating these massive libraries of mutants and growing them alongside the original, wild-type versions found in nature, they could precisely measure the growth rate of each variant. This provided a direct map of the fitness effects of thousands of individual mutations with a level of precision that was previously impossible. According to the neutral theory, the vast majority of these 12,267 mutations should have been either harmful or neutral. Beneficial mutations were expected to be so scarce that they might not even show up in a sample of this size. But the data told a different story. The Michigan researchers found that more than 1% of these amino acid altering mutations were actually beneficial. While 1% might sound small, in the context of evolutionary theory, it is an enormous, mathematically explosive number. It is orders of magnitude higher than the limits allowed by the neutral theory or even the nearly neutral theory. To put this in perspective, we can look at what this frequency of beneficial mutations would predict for the history of life. If 1% of mutations are advantageous and we apply the standard models of how genes become fixed in a population, we should see a rate of evolution that is blistering. Because beneficial mutations are favored by selection, they have a much higher probability of reaching fixation than neutral ones. If the raw material for adaptation is this common, these findings would predict that more than 99% of all fixed amino acid substitutions in our proteins should be adaptive. The molecular clock should not be a steady neutral tick, it should be a frantic race of constant improvement. Yet, when we look at the actual genomes of species, we do not see that frantic pace. We see a rate of change that looks, on the surface, remarkably like the slow neutral drift predicted 50 years ago. This creates a profound paradox. We have a high-speed engine of mutation running inside a vehicle that is only moving at a walking pace. If the raw material for adaptation is so common, why does the end result look so neutral? This discrepancy suggests that something is fundamentally wrong with our understanding of how beneficial mutations behave. Historically, we treated a beneficial mutation as a permanent upgrade, like a software patch that makes a program run faster and is never deleted. We assumed that once a helpful mutation appeared, natural selection would inevitably drive it to fixation until every member of the species carried it. But the high frequency of these mutations found in deep mutational scanning suggests that many upgrades are being generated, yet very few are being permanently installed. The discovery of these frequent beneficial mutations also forces us to reconsider the concept of the wild type. We often think of the common version of a gene found in nature as being optimized or perfected for its environment. If it were perfect, then almost any change to it should be harmful. However, the fact that Jianzi Zhang's team could easily find hundreds of mutations that make an organism grow faster suggests that natural populations are rarely, if ever, fully adapted to their surroundings. Instead, they seem to be in a state of perpetual chase. This brings us back to the original conflict between the Selectionists and the Neutralists. The Selectionists believed that the molecular world was a place of constant, fine-tuned adaptation, while the Neutralists argued it was a place of random noise. For decades, the Neutralists seemed to have the upper hand because the math of the molecular clock and the cost of selection made their model more plausible. But if the raw rate of beneficial mutations is actually high, then the Selectionists might have been right about the process, even if the Neutralists were right about the outcome. It suggests a world where natural selection is incredibly active, constantly pushing and pulling on thousands of variants, but where these forces often cancel each other out over long periods of time. This new data from deep mutational scanning provides a much more dynamic view of the genome. It is no longer a static blueprint where most changes are ignored. Instead, it is a highly sensitive system where a significant fraction of random changes can have a measurable impact on fitness. The high frequency of beneficial mutations suggests that the potential for evolution is much greater than we anticipated, but it also raises the question of what is holding it back. If life has such a ready supply of improvements, why has it not reached a state of perfection where beneficial mutations finally become as rare as the neutral theory predicted? As we look closer at these mutations, we find that the traditional boundaries between useful and useless DNA are blurring. The more we look, the more we find that the neutral parts of the genome are actually doing work, and the rare beneficial events are happening all around us. We are forced to ask if the appearance of neutrality in our genetic history is a true reflection of the mechanisms of life, or if it is merely a mask worn by a much more complex and active process of adaptation. If the neutral theory was built on the assumption that beneficial mutations are a biological impossibility at high rates, what happens to our understanding of our own history now that we know they are a biological commonplace? This tension between the abundance of potential adaptations and the slow reality of fixed genetic changes points toward a missing piece in our evolutionary puzzle. If a mutation can be a hero in one moment and a liability in the next, then the story of our evolution is not just a list of random accidents. It is a much more intricate drama. But if these beneficial mutations are so common, why do they almost never cross the finish line and become a permanent part of who we are? Chapter 2: The Moving Road and the Gambler's Ruin. The answer to why these genetic winners fail to reach the finish line begins with a mathematical reality that is as brutal as it is simple. Most beneficial mutations are dead on arrival. Even when a mutation provides a clear advantage, the 1% of hero mutations identified in recent scanning, it faces an immediate and terrifying hurdle known as stochastic loss. In the world of population genetics, this is the gambler's ruin. When a new beneficial variant first appears, it exists in only a single individual. Regardless of how much that mutation might improve the organism's fitness, the first individual carrying it is still subject to the whims of fate. They might be eaten by a predator, fail to find a mate, or simply have zero offspring by pure chance. Mathematically, the probability that a mutation will be lost in these first few generations is staggering. Even a mutation with a significant 1% advantage has a probability of loss defined by the relationship between its selection coefficient and the random drift of the population. For a new mutation with a 1% benefit, there is roughly a 98% chance that it will be extinguished within the first few generations simply by bad luck. The hero mutation is a tiny candle flame trying to stay lit in a hurricane of random events. Before natural selection can even see the advantage and begin to push the mutation through the population, the random sampling of reproduction often snuffs it out. This fragility is compounded by the fair coin of meiosis. In sexually reproducing organisms, an individual does not pass on their entire genome. They pass on only half. Every time an organism reproduces, there is a 50% chance that any specific gene, even a highly beneficial one, will simply not be included in the sperm or the egg. This random shuffling means that a beneficial mutation must win a series of coin flips just to survive its infancy. If it loses even once before it has spread to a handful of individuals, the evolutionary upgrade is deleted from history before it ever had a chance to prove its worth. Yet even for the mutations that manage to survive this initial stochastic gauntlet, the path to permanence is blocked by a more dynamic obstacle. To understand why advantageous mutations fail, we must stop viewing the environment as a static stage and start seeing it as a moving target. This brings us to the core of the paradox: adaptive tracking. Adaptive tracking is the process by which a population constantly shifts its genetic makeup to keep up with an environment that is in a state of perpetual flux. When the environment changes faster than the biological speed limit of fixation, the math of evolution breaks down. A mutation might begin its ascent, fueled by a sudden environmental shift that makes it valuable, but before it can reach every member of the species, the environment shifts again. The mechanism that turns these winners into losers is antagonistic pleiotropy. In this context, it means that the very same mutation that provides a massive advantage in one environment becomes a heavy liability in another. It is the hero-to-villain transformation of the genome. A mutation is not inherently good or bad. Its value is entirely dependent on the context of the world outside the cell. If the wind changes, the mutation that helped you fly might suddenly act as an anchor that drags you down. Researchers recently demonstrated this timed-out evolution using populations of yeast. By observing these organisms over 800 generations, with a new generation arriving every 3 hours, they were able to simulate the pressures of a changing world in a compressed time frame. In one arm of the study, yeast were kept in a constant, unchanging environment. As expected, beneficial mutations appeared and marched steadily toward fixation, eventually becoming the new standard for the population. But in the second arm, the researchers moved the yeast into a different type of growth media every 80 generations. They cycled through a variety of stressors, switching from glucose to galactose or maltose, moving from acidic to basic pH levels, and introducing high salt concentrations. The results were a perfect illustration of the adaptive tracking paradox. In the changing environment, beneficial mutations were appearing just as frequently as they were in the stable one, but they almost never reached fixation. A mutation that allowed the yeast to thrive in a high-salt environment would rise to 20 or 30% frequency, but then the researchers would shift the yeast into a low pH media, where that same mutation was harmful. Natural selection, which had been the mutation's greatest ally, suddenly became its executioner. The population was tracking the environment, but because the shifts happened every 80 generations, well before the hundreds of generations often required for a mutation to spread to the entire population, the mutations were constantly being suppressed or purged. If you were to look at the final genetic record of those yeast, the result would look remarkably neutral. You would see a slow, steady accumulation of changes that appeared to be the result of random drift. But this neutrality is a mask. It is the mathematical average of 1,000 failed adaptations. The yeast were not drifting, they were pedaling a bicycle as fast as possible on a road that was moving beneath them. They were working incredibly hard just to stay in the same place. This reveals that the steady tick of the molecular clock we observe in nature might not be the sound of random accidents, but the sound of a population frantically trying to keep its head above water in a world that changes faster than it can adapt. This has profound implications for human biology, particularly in how we understand the mismatch between our ancient genes and our modern lives. We often speak of the normal human genome, but adaptive tracking suggests we are carrying a genetic toolkit designed for a world that no longer exists. The most famous example of this is the thrifty gene hypothesis. For the vast majority of human history, our ancestors lived in environments where food was scarce and famines were frequent. In that context, mutations that allowed for highly efficient fat storage were hero mutations. They were the difference between life and death during a long winter or a failed hunt. Natural selection pushed these variants to high frequencies because they provided a massive survival advantage. However, in the modern world of high-calorie processed foods and sedentary lifestyles, those same hero mutations have become villains. The very genes that protected our ancestors from starvation now predispose us to obesity, type 2 diabetes, and heart disease. The environment shifted from one of scarcity to one of extreme abundance in just a few generations, far faster than the biological speed limit allows for a genetic correction. We are mismatched because our genomes are still tracking a world of the African savanna while our bodies are living in a world of supermarkets and office chairs. We are carrying the remnants of solutions for environments that have already vanished, and the new mutations appearing today that might help us adapt to our current world may never reach fixation before our culture and technology shift the goalposts once again. This perspective challenges the very definition of adaptation. We usually think of adaptation as a noun, a completed state where a species is fit for its niche. But if the environment is always in flux, then adaptation is a verb. It is a continuous, unfinished process of chasing a moving target. It suggests that the genetic variation we see within a species isn't just junk or random noise, but a reservoir of potential solutions. Some of these variants are the losers of the last environmental shift, while others are the potential winners of the next one. This also forces a reinterpretation of the molecular clock. We have long relied on the clock's steady tick to date the history of life, assuming that the rate of change is constant because it is driven by neutral drift. But if the rate of change is actually being driven by this constant cycle of selection and rejection, the clock is far more complex. The steady accumulation of mutations we see over millions of years is the residue of a high-stakes struggle where most beneficial changes are ephemeral. They are fleeting moments of brilliance that are extinguished by the next cold snap, the next drought, or the next migration. If even the most advantageous changes struggle to survive the whims of a changing world and the brutality of stochastic loss, then the parts of the genome we once dismissed as silent or junk might be carrying far more weight than we ever suspected. If a mutation can be a lifesaver in one generation and a death sentence in the next, then the neutrality we've observed for decades might just be an optical illusion caused by looking at life through too wide a lens. When we zoom in, we see that the battle for survival is happening at every single position in the genome, even in the places where we thought nothing was happening at all. This leads us to a new and even more controversial question. If silent mutations, those that don't even change the structure of a protein, can also be caught in this tug-of-war of selection, is there any part of our DNA that is truly, honestly neutral? Chapter 3. The Silent Code's Hidden Voice. For over 60 years, a specific type of genetic change has been treated as the ultimate proof of the neutral theory, the synonymous mutation. Often referred to in textbooks as silent mutations, these changes occur when a single letter in the DNA is swapped for another, but the resulting three-letter codon still calls for the exact same amino acid. Because the final protein remains unchanged, scientists long reasoned that these mutations were like changing the font of a word without changing its meaning. They were thought to be the perfect examples of pure neutral drift, invisible to the eyes of natural selection. In 1991, toward the end of his career, Moto Kimura pointed to the high rate of synonymous substitutions as the definitive proof of his theory. He argued that because these sites changed so much faster than those that altered proteins, they must be free from selective constraints. Ironically, the very evidence once used to prove the theory is what is now being reinterpreted as a signature of selection. The myth of the silent mutation began to crack with the observation of codon bias. If all synonymous versions of a gene were truly equal, we would expect to see them used randomly. Instead, across the entire tree of life, organisms show a distinct preference for certain codons over others. While this was once dismissed as a minor manufacturing optimization, a series of high-precision experiments is proving that these mutations are anything but quiet. In 2022, a research team at the University of Michigan, led by Jianzi Zhang, put this assumption to a definitive test. While Zhang's later 2025 research would look at amino acid-altering mutations across various species, this 2022 study focused specifically on the synonymous landscape of yeast. Using CRISPR technology, they induced hundreds of synonymous mutations and measured the actual fitness of the resulting strains. The results were a seismic shock. If the neutral theory were correct, these mutations should have had a fitness effect of 0. Instead, the researchers found that a staggering 76% of these synonymous mutations were significantly deleterious. They weren't just neutral, they were harmful. In many cases, these silent changes were just as damaging as mutations that actually altered the protein structure. Only about 1.3% were beneficial. This finding suggests that the assumption that synonymous sites provide a safe harbor for neutral drift is fundamentally flawed. However, Zhang noted a crucial caveat. These results were observed in unicellular yeast, and while the implications are profound, the exact proportions of these effects must still be confirmed in more complex multicellular organisms. How can a mutation that doesn't change a single amino acid be so destructive? The answer lies in the realization that the genetic code is an intricate set of logistical instructions. Synonymous mutations can disrupt every intermediate step between a gene and a finished protein. The first of these is the actual quantity of the message produced. The 2022 yeast study emphasized that the primary driver of deleterious effects was often a reduction in the transcript level, the actual amount of messenger RNA, mRNA, the cell created. It wasn't just that the message was bad, it was that the cell wasn't making enough of it to function. While the stability of the mRNA molecule, how it folds and how long it lasts, is also a factor, the sheer volume of production appears to be the critical bottleneck. The second major hurdle is the speed of construction. The cell uses tRNAs to bring amino acids to the protein-building machinery. If a synonymous mutation swaps a common codon for a rare one, the process can suffer from ribosome stalling or translational pausing. This isn't merely a delay, it is a structural hazard. Proteins begin to fold into their complex three-dimensional shapes while they are still being built. If the construction process pauses at the wrong moment because of a slow codon, the protein might tangle itself into a useless knot. There is a hard physical limit to this flexibility. Research by Jordi Vila and others has identified a specific physical constraint, a free energy threshold of approximately 7.4 kilocalories per mole for protein stability. Proteins exist on a knife's edge. If a synonymous mutation alters the timing of folding or the stability of the mRNA, it can push the molecule past this threshold, causing it to unfold or aggregate into toxic clumps. In biology, timing and stability are not just details, they are matters of survival. This discovery points toward a second code hidden within our DNA. While the first code tells the cell which ingredients to use, the second code governs the speed, volume, and folding of the process. This second code is under intense pressure from natural selection and is linked tightly to epigenetic markers, chemical tags that don't change the sequence but dictate how the sequence interacts with the cell's folding and repair machinery. When we see codon bias in nature, we are seeing the result of millions of years of selection, weeding out the synonymous mutations that were legal in the first code, but illegal in the second. This sensitivity extends to how the genome manages future risks, a concept known as codon volatility. Some codons are more dangerous than others because they are only one mutation away from a much worse amino acid. Consider the arginine codon CGG. If it mutates, its mutational neighbors, the amino acids it is most likely to become, include tryptophan or proline, which can be structurally disruptive. However, if the cell uses the synonymous arginine codon AGG, a single letter change would instead lead to lysine or serine. The meaning of the word is currently the same, but the consequences of a future typo are entirely different. Genes under intense selection often use codons that are less volatile, a form of genomic risk management that minimizes the damage of future mutations. The implications for human health are immediate. For decades, synonymous mutations in patients were dismissed as harmless polymorphisms. We now realize these silent changes are likely hidden drivers of disease, contributing to everything from cancer to neurological disorders by altering protein levels or folding pathways. Because these sites were used to calibrate the molecular clock, their non-neutrality suggests our estimates for species divergence may be fundamentally skewed. As we peel back the layers of the silent mutation, the idea that we can swap out parts of our DNA without consequence is fading. If the genome is this sensitive to even the smallest changes, and if harmful mutations are this common, how does life manage to protect its most vital functions? If the silent parts of our code are under such heavy guard, what does that tell us about the regions of the genome that don't code for proteins at all? Chapter 4. The Shielded Genome. For as long as we have studied the molecular basis of life, we have operated under a nearly universal assumption that mutation is a blind, random process. We have been taught to visualize mutations as a genetic hailstorm, randomly pelted against the roof of an organism's DNA. In this traditional view, the storm doesn't care whether it hits a vital structural beam or a decorative shingle. It is the job of natural selection to act as the repair crew, surveying the damage after the storm has passed and deciding which holes must be patched and which can be ignored. This randomness was the very heart of the neutral theory. If mutations occur with total indifference to their location, then the patterns we see in our genes are simply the result of which random hits were lucky enough to survive the filter of selection. But as we peer deeper into the actual architecture of the genome, we are discovering that the storm isn't random at all. Life has found a way to hold an umbrella over its most essential parts. This discovery of non-random mutation represents one of the most significant shifts in evolutionary biology in decades. It suggests that the genome is not a passive target for errors, but an active manager of its own mutational risk, a concept that fits within the framework of biological evolution cybernetics. This view treats the genome as an automatic control process, a system that has evolved to govern its own variation. This governance is not a conscious decision by the DNA, but a physical result of biochemical limits and selective constraints that have been honed over billions of years. To understand how this works, we have to look at a humble flowering weed called thale cress. Recently, researchers conducted a massive analysis of hundreds of different lines of this plant, sequencing their entire genetic libraries to track exactly where mutations were occurring. If the assumption of randomness were correct, these mutations should have been scattered evenly across the plant's chromosomes. Instead, the data revealed a highly structured and biased pattern. Mutations were significantly less likely to occur in the regions of the genome that were biologically essential. These high-value areas, the genes responsible for fundamental cellular processes like growth and development, appeared to be under a form of high-tech protection. These protected regions are known as mutation cold spots. In these areas, the mutation rate is significantly lower than in the rest of the genome. It wasn't just that selection was removing the bad mutations after they happened. The mutations were simply being prevented from occurring in the first place. The mechanism behind this protection is a masterclass in biological engineering. Our DNA is tightly wound around proteins called histones, forming a complex structure known as chromatin. In Thaleocress, and likely in many other organisms, the most essential genes are wrapped in a specific way that makes them less accessible to the chemical and physical forces that cause mutations. This is governed by epigenetic markers, chemical tags that signal the cell's repair machinery to prioritize these essential regions. This brings us to the second layer of the defense system, targeted DNA repair. The cell's repair machinery is specifically recruited to active essential gene regions. By focusing its limited resources on the parts of the genome that the organism can't afford to lose, the cell effectively lowers the mutation rate in those areas, while allowing the rest of the genome to fluctuate more freely. This is a form of evolutionary risk management that the neutral theory never accounted for. Because the mutation rate is a moving target, low in essential genes and high in peripheral ones, our molecular clocks are ticking at different speeds in different parts of the same genome. The regions we thought were evolving slowly because of intense selection might actually be evolving slowly because they are being protected from mutation in the first place. However, this protection is not perfect, and its effectiveness varies across the tree of life. This variation is explained by the drift barrier hypothesis. Natural selection always tries to lower the mutation rate to protect the organism's integrity, but it hits a barrier set by genetic drift. In large populations, such as those of many bacteria, selection is highly efficient and can push the mutation rate very low. In smaller populations, like humans, the power of random drift is much stronger, making selection less effective. As a result, our genomic protection is leakier than that of a bacterium. We are forced to tolerate a higher background mutation rate because our population size doesn't provide selection with enough power to lower the barrier further. This leakiness is further complicated by a biochemical quirk known as biased gene conversion, BGC. In mammals, the process of gene conversion, where DNA sequences are swamped and repaired, is biased. Specifically, the machinery is more likely to pass down GC base pairs than AT base pairs. This is a non-random biochemical process that has nothing to do with the fitness of the organism, yet it makes neutral mutations behave as if they are under selection. Because GC pairs are favored, they spread through the population more quickly than they would by random drift alone. This process mimics the signal of positive selection, creating an illusion of adaptation where there is only a mechanical bias. When we combine the effects of targeted protection, the drift barrier, and biased gene conversion, the quantitative punchline is striking. Researchers now estimate that because of genetic hitchhiking and BGC, only about 5% of the human genome is truly evolving neutrally. The rest is either under direct selection, protected by repair mechanisms, or swept along by biochemical biases. The neutral background noise we've been measuring for 50 years is actually a highly curated signal. This concept of mutation bias and genomic governance extends into the world of prokaryotes, but it manifests in different ways depending on the lineage. Recent large-scale genomic analyses of over 22,000 bacterial and archaeal genomes have revealed that these organisms follow different selective regimes. In many bacterial groups, there is a clear signature of selection for functional diversification. As these bacteria grow larger genomes, they face stronger selection pressure to maintain that expanded toolkit. The more tools they have, the more precisely those tools must be managed. In contrast, archaea show a much stronger signature of pure streamlining. In these ancient organisms, larger genomes actually correlate with weaker selection. In their world, the pressure is almost always to keep the genome as small and efficient as possible. This heterogeneity tells us that there is no single law of molecular evolution. The neutral theory attempted to provide a one-size-fits-all explanation, but the reality is a patchwork of different strategies. Some organisms manage their genomic risk by protecting essential genes. Others manage it by ruthlessly deleting anything that isn't essential. This leads us to a startling realization about the nature of non-coding DNA. For decades, we were told that the vast majority of our genome was just a graveyard of dead genes and useless repeats. But if the genome is actively directing mutations toward these non-essential regions to protect the essential ones, then that junk is serving a vital purpose. It acts as a mutational buffer, a genetic crumple zone that absorbs the inevitable errors of life, keeping them away from the high-value targets. In this light, non-coding DNA isn't just an accident of history. It's a strategic defense. The accumulation of this DNA might actually be an active adaptation to preserve genomic integrity. This also forces us to reconsider the role of the environment. We used to think that the environment only acted as a filter after mutations occurred. But environmental stress can actually trigger changes in how the genome protects itself. When an organism is under intense pressure, the cell's epigenetic markers can shift, temporarily relaxing the protection on certain regions. This allows for a localized burst of variation that could help the organism find a solution to its problem. It is a way for life to tune its own evolutionability, allowing the genome to be conservative in stable times and take risks in times of crisis. This level of control over the mutational process challenges the very definition of a neutral change. If the cell's own machinery is steering the hailstorm of mutation, then every single change in the genome is, in some sense, a product of the organism's evolutionary history. We are moving toward a view where the neutral background of our DNA is actually the result of an incredibly ancient and sophisticated system of genomic governance. This governance might even contribute to the resolution of classical paradoxes of variation, suggesting that the stability we see in large populations is not a lack of potential, but a highly evolved state of defense. The evidence of non-random mutation suggests that life has been gaming the system for billions of years. By directing the inevitable errors of replication toward the regions where they do the least harm, and by focusing repair efforts where they matter most, organisms have created a molecular world that looks neutral on the surface, but is actually highly managed. We aren't just the products of the mutations that were selected, we are the products of the mutations that were prevented. As we recognize that the genome is an active participant in its own evolution, we have to ask how this changes our ability to quantify the true scale of adaptation. If the background noise isn't random, then every tool we've used to detect selection might be giving us a distorted picture. We have spent half a century trying to find the signal of adaptation buried in a sea of neutral noise, only to find that the noise itself is a curated product of evolution. If the neutral theory can no longer serve as a simple, reliable null hypothesis, how do we begin to measure the true extent of life's struggle to adapt? Chapter 5. The Selectionist Resurgence, Quantifying the Adaptive Surge. We have reached a moment in biological science where the ground beneath our feet is shifting. For decades, the neutral theory was the quiet, steady heartbeat of evolutionary biology, providing a simple, elegant way to measure the history of life. But today, we are witnessing a powerful selectionist resurgence. This isn't just a minor correction to an old idea. It is a fundamental reconstruction of how we view the molecular world. We are moving away from a view where the genome is a vast, silent ocean of random drift, and toward a vision of the genome as a high-stakes arena where natural selection is the primary architect, working at nearly every position in the genetic code. However, as we embrace this more active view of evolution, we are faced with an incredibly difficult challenge. If selection is everywhere, how do we actually measure it? How do we quantify the true scale of adaptive evolution when the evidence is so often hidden by the very processes that create it? The difficulty of this task is best illustrated by the wildly different answers scientists get when they try to measure the adaptive fraction of a genome. This is the percentage of amino acid changes that were driven to fixation by positive, beneficial selection rather than random drift. When researchers apply different statistical tools to the same species, the results are often bafflingly inconsistent. In some studies of various plants and animals, the estimate for adaptive evolution is nearly zero, suggesting the neutral theory still holds the crown. But in other studies, especially those focusing on high recombination species like fruit flies, the estimate for adaptive evolution shoots up to 90%. This massive range, from nothing to nearly everything, reveals that our current tools are struggling to capture the full complexity of the molecular struggle and the true intensity of the selective forces at play. One of the primary tools in this effort is a statistical method that compares the patterns of variation within a species to the differences between species. The logic is straightforward. If most evolution is neutral, then the ratio of synonymous to non-synonymous changes should be roughly the same whether you are looking at two individuals in a population or comparing a human to a chimpanzee. But if natural selection is actively favoring certain protein changes, we should see an excess of non-synonymous substitutions in the long-term record between species. While this method has been a workhorse for decades, we are now realizing it has a major blind spot. It assumes that neutral and selected sites are independent of one another. But in the densely packed and highly interactive world of the genome, nothing is truly independent. This brings us to the phenomenon of linked selection, which is perhaps the greatest challenge to quantifying the scale of evolution. Selection doesn't act on a single letter of DNA in isolation. It acts on the entire physical chunk of the chromosome where that mutation sits. When a beneficial mutation is driven rapidly to fixation by natural selection, a process known as a selective sweep, it acts like a genetic vacuum, dragging every nearby neutral mutation along with it. Conversely, when selection removes a harmful mutation, it often takes out the surrounding neutral variation as well, a process called background selection. This physical linkage means that the fate of a neutral mutation is often decided not by its own properties, but by the company it keeps. This creates a massive problem for scientists trying to find the signal of adaptation. Because linked selection can mimic the appearance of neutral drift, it can mask the true extent of the selective forces at play. Recent ambitious studies have attempted to account for these neighborhood effects by looking at how genetic variation changes in relation to recombination rates. In regions of the genome where DNA is frequently shuffled and swapped during reproduction, neutral mutations can break free from their selected neighbors and behave according to the laws of drift. But in regions with low recombination, the hand of selection is felt everywhere. When researchers applied these more sophisticated models to the human genome, they reached a startling conclusion. Only a small fraction of our entire genetic library can be truly classified as evolving neutrally. The vast majority of our genetic code is being influenced, directly or indirectly, by the relentless and pervasive pressure of selection. This realization helps solve one of the oldest mysteries in the field, known as Lewontin's paradox. For a long time, scientists couldn't understand why large populations, like those of insects or marine invertebrates, didn't have vastly more genetic diversity than small populations, like humans. Neutral theory predicted that diversity should scale linearly with population size, but it clearly didn't. The selectionist resurgence provides the answer. In large populations, natural selection is so incredibly efficient that it is constantly cleaning the genome. Selective sweeps and background selection are happening so frequently that they are systematically wiping out the neutral variation that would otherwise accumulate. The lack of diversity in these species isn't a sign of neutral drift. It's a sign that natural selection is so powerful it has reached a state of genomic dominance, effectively overriding the effects of population size. This shifted perspective is now being bolstered by the tools of the 21st century, specifically machine learning and the study of complex systems. Researchers are beginning to view protein evolution not as a simple linear progression, but as a complex system with sensitivity to initial conditions. This means that a single mutation early in a species' history can fundamentally change the pathway of all future evolution, a concept known as epistasis. A mutation that is beneficial today might only be possible because of a seemingly neutral change that happened a million years ago. This interdependency makes it even harder to quantify selection, because the value of a mutation is constantly being recalculated based on the rest of the genome and the historical context of the lineage. Machine learning models are now being trained to recognize the subtle, nonlinear signatures that these complex selective forces leave behind. They are finding that natural selection often works through polygenic adaptation, where hundreds of small shifts in the frequencies of many different genes happen simultaneously. This is much harder to detect than a single dramatic selective sweep, but it may be the primary way that complex traits like intelligence, height, or immune resilience actually evolve. We are moving away from looking for the gene for a trait and toward looking for the genomic signature of a population, shifting its entire balance toward a new environmental target, a process that requires a holistic view of the entire genetic landscape. The Selectionist resurgence also forces us to rethink the very energy of life. Recent perspectives have suggested that mutation itself is the driving force of evolution at both the genic and phenotypic levels. This mutationism doesn't replace selection, but it argues that the specific mutations that arise and the order in which they appear dictate the direction of life just as much as the environment does. Because gene duplication and other large-scale DNA changes occur more or less at random, the menu of options available to natural selection is limited by chance. This adds a layer of fortuitous randomness to evolution that is different from neutral drift. It's not that the changes don't matter, it's that the choices available to selection are themselves a product of a random mutational lottery, where the house always has a say in what can be played. This brings us to a new understanding of the units of evolution. We are finding that most phenotypic characters are controlled by the interaction of massive multigene families that evolve through a birth and death process. Genes are constantly being duplicated, modified, and then either put to work or allowed to wither away into pseudogenes. The neutral theory saw the existence of these duplicate genes and junk sequences as proof that the genome was mostly a graveyard of random accidents. But the Selectionist resurgence sees this as a vibrant, active nursery. These extra copies provide the excess capacity that allows a lineage to experiment with new functions without risking its survival. What looked like neutral bloat to a scientist 50 years ago now looks like the essential research and development department of a successful species, providing the raw material for future innovation. As we integrate these findings, we are seeing the emergence of a more unified theory of molecular evolution, one that recognizes the power of drift in small populations and the dominance of selection in large ones, but places both within the context of an ever-changing world. We are beginning to see that the neutralist-selectionist debate was a necessary conflict that allowed us to develop the rigorous tools we have today. We no longer have to choose between a world of random noise and a world of perfect adaptation. Instead, we see a world where randomness and selection are inextricably linked. Random mutations provide the raw material, but selection is the force that decides which parts of that material are allowed to be neutral and which must be held under strict control, creating a delicate balance between stability and change. This new vision has profound implications for how we approach human health. If the vast majority of our genome is under some form of selection, then there is virtually no such thing as a safe mutation. We can no longer ignore the non-coding regions of our DNA when we look for the causes of disease. We are realizing that our susceptibility to illness is written in the subtle variations that we once dismissed as noise. We are the products of a system that is far more sensitive and far more active than we ever dared to believe, where every letter of the code has the potential to influence our biological destiny. The challenge of quantifying the true scale of adaptive evolution remains the great frontier of modern biology. Every time we think we have found a way to measure the signal of selection, we discover a new layer of complexity. Linked selection, biased gene conversion, or shifting environmental pressures that complicates the picture. We are like astronomers trying to map the stars through a thick moving fog. We know the stars are there, and we can see their light, but their exact positions and numbers are constantly being distorted by the atmosphere of the genome itself, requiring ever more sophisticated lenses to see clearly. This leads us to a final, provocative realization. If the neutral theory was a simplification that allowed us to build the first maps of the genetic world, the selectionist resurgence is the high-resolution satellite imagery that is showing us how much we missed. We are finding that the neutrality of life is often a byproduct of the sheer complexity of the genomic landscape. We are seeing the results of 1 billion tiny battles, won and lost at every position in the code, every single day, creating a tapestry of life that is far more intricate than we once imagined. As we stand at the edge of this new era in genomics, we are forced to ask, if the simple, neutral models of the past are no longer enough to explain the vibrant complexity of the DNA we are now sequencing, what will the next generation of evolutionary theory look like? If we can no longer rely on the molecular clock as a simple, constant measure of time, how will we reconstruct the history of our own species? And if the vast majority of our genetic variation is not just random noise, but a living, shifting record of our ongoing struggle to survive in a world that never stands still, what does that tell us about the hidden potential for our own future evolution and the resilience of life itself? In conclusion, the neutral theory of molecular evolution is being fundamentally challenged. We now see that beneficial mutations are far more common than once assumed. Silent changes are not neutral, and genomes actively direct mutations away from essential genes. This reveals a world where natural selection is pervasive, shaping most genetic changes. These insights force us to rethink adaptation, the molecular clock, and even the causes of disease. As we move beyond simple models, we uncover a more complex and dynamic evolutionary process, where life's history is written not in random drift, but in the relentless push and pull of selection. Thank you for joining this exploration.